My name is Sheetal Gandotra and I'm working on understanding how lipids that are stored within macrophages are mobilized or released during infection so that Mycobacterium tuberculosis, which is an intracellular pathogen, can gain access to these. So we know that macrophages, like many cells, are able to store excess fatty acids into organelles which are called as lipid droplets. Now these lipid droplets are storage of fat, but they're not just that. There's actually a lot of proteins that sit on the surface of these lipid droplets, and the amount and the quality or the type of proteins that localize to these droplets change as a result of the stimulus that is given to the cells. And what we've seen in an earlier study is that a protein called R-late-B, which is an otherwise lysosomally localized protein, localizes to these lipid droplets. So we want, and the abundance of this protein on the lipid droplet changes as a result of MTB infection or Mycobacterium tuberculosis infection. So in this project, we're trying to understand the role that this protein plays in the mobilization of lipids during infection. So what we're trying to do here is, one, understand what happens to the mobilization of lipids if R-late-B is not there or is present in an altered form. How does R-late-B actually localize to the lipid droplet is one of the important questions that we're trying to address here. And that's where the skill sets that my lab has and the skill sets that my collaborators' lab in Paris has are really going to complement each other in understanding this regulated localization of a protein onto the lysosome or to the lipid droplet. This project is really an opportunity for people working in my lab, which is an infection biology lab and a lipid droplet biology lab, to interact with a lab that studies lipid droplets from the context of biophysics. And here we have a protein of choice that came up from the infection studies, and now we want to address the role of this protein in how it binds to lipid droplets and what impact it has on lipid mobilization processes. So together, with this teamwork, what we expect to come up with are ways by which a macrophage is able to mobilize its lipids. And I think the impact of this work is not going to be limited only to MTB infection, but possibly many other pathological situations where lipid accumulation in macrophages is deregulated. So this, I think, is going to be the larger impact of the work that we're doing within this project. Cells store lipids and release them in a regulated manner is the underlying feature of several pathologies. Whether you look at atherosclerosis, whether you look at fat accumulation, even in morbid obesity, whether you look at it in terms of infections like tuberculosis. So my current interest is in the area of tuberculosis, but the research that we do pertains to lipid mobilization and lipid storage within macrophages. And because this is a central process in macrophages and it is involved in regulating the inflammatory processes as well, I think this project can have huge impact on the way we think about how macrophages can be tuned or be corrected for the pathologies that they're involved in. So I think that's the longer term contribution that this project would make in the area of science. CEFIPRA, being a bilateral grant between a lab in India and a lab in France, really helped me bridge the gap that we had in our research, taking it to the level of understanding biophysical aspects of how a protein would localize to the lipid droplet. And this is actually humongous when you think about the kind of exponential jump that a project can make by bringing together people of different expertise together. And I think by finding the right partner through CEFIPRA and getting this grant, being able to work together has become possible. And I think we would like to take this forward in building this relationship of two investigators working on lipid droplet biology even further. And I think it's going to impact the projects that my lab does and the projects that my collaborators' lab does, and which is not going to be restricted only to the project that we do today.
Hi, my name is Dilip Menon. I am currently a PhD student at CEFIPRA IGIT, and I am working with Dr. Sheetal Gandotra. So I was part of the CEFIPRA project, which involved a collaboration between our lab and a French lab, who was French lab, Dr. Abdou Rachid Thiam's lab. So the biggest advantage as a PhD student I had from this particular project is that we have the option of collaborating with an international PI who was an expert in a field which was close by us but had different skill sets. Thereby, I had this opportunity to learn new things which otherwise would have been restricted to us, and this is the biggest advantage I guess I had from this project.
Long live India and France friendship.